Hello, my name is David Dominguez and I am an animal science major, and today I'll be talking about finding if the slick allele is in Brangus cattle and if it affects their hair length. So thermal tolerance is a very important part of this entire experiment. It's a term used to describe if an organism can live and thrive under high temperatures. Some breeds are far better at this than others. An example of that are Brahman. They are more adapted to hotter climates through evolution and breeding. Other breeds actually have genetic mutations that allow them to have shorter, sleeker coats. An example of this are Holsteins. Holsteins actually have the mutation known as SLIC. SLIC is an allele of the prolactin receptor PRLR gene that causes cattle to have shorter, sleeker coats. So the hypothesis of this entire experiment was that the cattle from the Seminole tribe that were collected would not show any signs of the SLIC allele, and they would just show up as wild type. So 117 samples were collected from 2016 to 2017 from Brangus cattle from the Seminole tribe in Okeechobee, Florida. The blood samples had their DNA extracted, and also hair samples were taken to be measured for length in millimeters. The DNA extraction, the DNA itself went through PCR, and then after PCR went through an enzyme digest, and then after that it was put into an electrogel phoresis. So to see how long the base pairs were, to see if it actually had the SLIC allele, or the DNA that coded for the SLIC allele. So they were all the cattle were put into three different categories. There were wild type, heterozygotes, and mutants. Out of the 117 samples, there were 12 heterozygotes, and there were 105 wild types and no mutants. So there was a numerical difference, but after using the program R, there was no statistical significant difference between hair length and the heterozygotes and wild types. Actually, you can see that in Table 1, the numerical difference was that in the hets actually had shorter hair in the longer hair category and even shorter hair in the shorter hair category compared to their wild type counterparts. So in total, in conclusion of this experiment, the hypothesis did not support what I found. We actually did find 12 heterozygotes. And since there was no statistical significance in the data, that means that we cannot detect the effect of the mutation of the SLIC allele. But since we have heterozygotes, that means that there's a possibility of a mutant out there. So if I were to redo this experiment again, the total number of samples used would be increased, and to get a broader picture of the whole thing and to see if there actually is a mutant in the Seminole tribe. So thank you and have a wonderful day.